Hey everybody, if you're just joining us right now, we're going to give it a minute or so until all registrants get in here, and then we'll be on our way shortly. Thanks for your patience. All right, hang on one second. All right, I think we've arrived. Hopefully you can see that screen. Welcome and thanks everybody for joining us on our 13th episode of Ewing's U. And sorry for the delay on our end. I was just looking back at our last episode and it was in July. But just know that these sarcoma experts are few and far in between, and we will do our best to schedule them as they appear. So stay patient with us. Thank you. Anyways, for those of you that don't know me, I'm Piero Spada. I'm going to be your co-host once again today. I'm also co-founder at Little Warrior Foundation, and more importantly, father to now 13-year-old little warrior Lucy. If you're part of our core contingency, aka the Little Warrior Fight Club, thanks for being here with us since day one and making this a place for positive change. And if you're new, well, welcome to Ewing's U. It's our webinar series where we hear from leading experts and discuss the cutting edge care and the science behind Ewing sarcoma. On that note, today we'll be hearing from one prominent expert in the field, Dr. David Loeb, who is one of the few experts that wears two hats as both a clinician as well as a researcher in this field. Specifically, today we'll be exploring a novel means to prevent Ewing sarcoma metastases from occurring through the use of a drug called DFMO. Briefly, Dr. Loeb graduated from Johns Hopkins University with a Bachelor of Science and then obtained his MD and PhD from Columbia University. He then returned to Johns Hopkins, where he completed both his residency in pediatrics, his fellowship in pediatric hemonc, and then later joined the faculty at Johns Hopkins, where he practiced and performed research for 17 years. Dr. Loeb then joined the faculty at Children's Hospital in Montefiore, where he currently serves as chief in the Division of Pediatric Hemonc and Cellular Therapy. He is the director of the sarcoma program there and also a professor of pediatrics and developmental and molecular biology at the affiliated Albert Einstein College of Medicine. On the research side, Dr. Loeb has an active translational research laboratory focused on understanding bone tumor metastases. His laboratory developed a clinically relevant mouse model of sarcoma metastasis and has used this model to perform preclinical testing of novel agents that can interfere with this process. More basic scientific studies in the lab involve exploring the role of Wnt signaling pathway in Ewing sarcoma migration, invasion, and metastases. And Dr. Loeb is also studying the role of an RNA helicase enzyme, aka DDX3, in Ewing sarcoma biology, especially how this enzyme affects the repair of damaged DNA. More recently, the laboratory has developed an interest in targeting the metabolic reprogramming associated with the metastasis as a way to prevent the outgrowth of distant metastases from disseminated tumor cells. Lastly, Dr. Loeb is also actively involved in a clinical research involving the development of radiopharmaceutical agents for the treatment of bone metastases and the development of a small molecule inhibitor of DDX3. He serves as the local PI for a clinical trial of reduced intensity haploidentical bone marrow transplantation for children with high risk solid tumor. Finally, as an offshoot of his laboratory work, Dr. Loeb is involved in the development of biomarkers of metastatic risk and of mean residual disease in children and AYA population. But before we kick things off with him, I just want to remind everybody that Ewing's U is a webinar production of the Little Warrior Foundation. We are a national childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out $1.6 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us at Little Warrior Foundation to make programs like this a reality, we rely on the support of little and big warrior communities alike. We appreciate all of your support so we can continue our mission of bulldozing our way towards more effective and less toxic therapies and continue this vital work for kids battling Ewing sarcoma. With that, I'm going to hand it off to my omnipresent co-founder and co-host, Emily, to introduce herself and touch on a couple of housekeeping items.
Hi, everyone. I'm Emily McFadden, one of the co-founders of Little Warrior Foundation. I'll be help moderating today. Please use the chat function in your Zoom to ask questions, or sorry, the Q&A function to ask questions, but feel free to also use the chat functionality. Dr. Loeb has specifically asked for lots of feedback from the patient community today, and would love to hear from you in the chat. So we'll be checking both places as Dr. Loeb talks, and at the end, excuse me, we'll ask everybody's questions. As always, we can't discuss specific cases in the questions or provide direct medical advice through this webinar, but we hope to have lively discussion. And as always, we will be recording this session to be posted to our blog later this week, so you can rewatch or share with someone who might find this information valuable. With that, I will pass it over to Dr. Loeb.
Well, thank you so much for inviting me to participate and to talk about this project that we're involved in, and for that overly generous introduction that seemed to go on forever that would really make my mother proud. I want to start by sharing the screen and hopefully sharing the correct screen. Hopefully you guys see a title slide. Yes, perfect. All right, so I'm going to talk about a project in our lab that involves using a drug called DFMO to attack a particular metabolic pathway called the production of polyamines, and how we think this can inhibit Ewing sarcoma metastasis through inducing a relatively understudied type of cell death called ferroptosis. So I'm sure you guys all know the basic background information on Ewing sarcoma, which is the second most common bone tumor in kids. This is what a plain X-ray of a leg involved with Ewing sarcoma looks like. The disease predominantly of adolescents and young adults, a little bit more common in boys than in girls, a little bit more common in some bones in the skeleton than others, driven by a chromosome translocation between chromosomes 11 and 22, predominantly. The most important thing to keep in mind about the chromosome translocation is that although we know it regulates the expression of lots of different genes, it's not actually really clear exactly what the key events are that cause cells to become Ewing sarcoma cells just because this happens. And despite tons and tons of work at trying to identify characteristics of patients who are likely to do well compared to patients who are not likely to do well, the most important prognostic factor remains whether you have metastasis or don't have metastasis. And so a few years ago, we decided that maybe one of the reasons that we haven't been making as good progress in treating Ewing sarcoma as most of us would like is because so much preclinical work is focused on doing a better job of killing primary tumors and not so much focused on understanding how metastasis happens and what we can do about that. And since metastasis is the most important factor in determining who's going to survive and who's not, we decided that we would focus on that. Which raises the question, how do you study metastasis? Because you can't just put cells on a dish in your incubator in your lab and study metastasis that way. And so we set about to develop what I refer to as a clinically relevant mouse model of spontaneous metastasis. And most of this work was done by the gentleman in the picture there, Seth Goldstein, who is now a pediatric surgical oncologist at Northwestern in Chicago. But he spent some time in my lab and developed this model. It's different from the way most labs study metastasis. Most labs use what's called tail vein injection model, which essentially means you take the cells that you're interested in, you inject them into the tail vein of the mouse, and they disseminate through the mouse's body, land where they're going to land, and grow into metastases. And that's useful for studying how cells in the bloodstream can interact with distant organs, but it really doesn't capture the whole spectrum of what happens when a human patient with Ewing sarcoma undergoes metastasis. And so we do something that's a little bit different, and I apologize for the icky picture of a mouse undergoing surgery and then recovering afterwards. But we essentially developed this model that more closely mimics what happens to actual patients. So we take a fragment of Ewing sarcoma, either grown from a patient or grown in a donor mouse, and we surgically implant that fragment of tumor into the tibia of the mouse, and then we sew the mouse's leg back up, and we let the tumor grow in the leg where it belongs. When the tumor gets to be an appropriate size, we do a second surgery where we amputate the affected limb but leave the mouse alive. And when we do this, eventually the mice will develop metastases and then ultimately die from the metastatic disease that they develop. And this more closely mimics what happens in humans, because nobody injects cancer cells into the bloodstream of a human to develop metastases. They grow from a tumor that's growing in the bone, and the primary tumor is addressed with surgery or radiation, chemotherapy, or a combination of those things. But the metastases form from that primary tumor and spontaneously spread, and that's what we see in our model. And some important things to note is that this really does reflect an interaction between the primary tumor and its environment, because if we take fragments of various different tumors, and this is data from our paper that included Ewing sarcoma, osteosarcoma, and rhabdomyosarcoma, but if we take these tumor fragments and we implant them under the skin of the mice, they never metastasize. You see the row of zeros over here. But if we take exactly the same tumor fragments and implant them in the bone instead of in the subcutaneous space, we do see metastases, and the percentage of mice that develop metastases can range from 25% up to 90% depending on the model, and that sort of reflects what happens in real humans too, where some patients have more aggressive disease and other patients have less aggressive disease. We have tumor models that are more aggressive and less aggressive. But the take-home message here is that the environment where the tumor is growing matters. And we can see that in this picture here that leads to why we are thinking about polyamines and DFMO in the first place. So here you can see tumor cells growing in close proximity to normal bone. Here it's tumor cells growing in and around muscle fibers to show that when we surgically implant the tumor into the correct microenvironment, the cancer cells and the normal cells are able to physically interact with each other, and that affects the biology of the tumor. And so one of the things that we found early on is that this enzyme called arginase is expressed at much higher levels in subcutaneous tumors than in orthotopic tumors, meaning implanted into the bone, even though both tumors originated from the same donor mouse, which means that the biology of the tumor is being influenced by where in the mouse it's growing. And arginase was one of the things that we looked at. So what's so special about arginase? Arginase is a key component of one of the metabolic pathways in most cells called the urea cycle, which is the major way that cells deal with waste nitrogen when they break down proteins. Arginase is here in the urea cycle, and it metabolizes the conversion of an amino acid called arginine into a different compound called ornithine. Ornithine is converted by another enzyme called ODC, which stands for ornithine decarboxylase, into compounds called polyamines. So what are polyamines and what do they do? Polyamines are low molecular mass, highly positively charged molecules that have been implicated in lots and lots of processes in other tumor types, and I listed a bunch of them here, and you'll see that metastasis is on the list. There are three main species, and they're called putrescine, which the chemical formula is here, and spermidine, the chemical formula is here, and spermine is here. Targeting polyamines in cancer therapy is actually not a new idea for us. It's actually been something that's been studied extensively in colorectal cancer, in neuroblastoma, and this is really important. DFMO, the compound that I'm going to talk about, has been studied specifically as a maintenance therapy in patients with high-risk neuroblastoma, and it's likely that the FDA is going to approve DFMO as maintenance therapy for high-risk neuroblastoma patients before the end of the year, based on the results from this and other studies. DFMO is an irreversible inhibitor of ODC. ODC is the rate-limiting enzyme in polyamine biosynthesis. So DFMO targets ODC and prevents polyamine synthesis. So we started to think that if a difference in expression of arginase, which is right upstream of ODC, can differ between subcutaneous tumors that don't metastasize and tumors implanted into the leg which do metastasize, then maybe polyamines, which are downstream from that, since they've been implicated in metastasis, are important in Ewing sarcoma metastasis. So we started to investigate the role of polyamines in Ewing sarcoma metastasis and the ability of DFMO to affect various aspects of Ewing sarcoma biology. So the first thing we looked at was the ability of DFMO to impact Ewing sarcoma proliferation in vitro. And this was work that was started by this gentleman, Kyle Jackson, who's currently on the faculty at Indiana University, and this woman, Rachel Offenbacher, who is currently a junior faculty member at Einstein and Montefiore with me and still working in my lab. And what we were able to show is in 3 different Ewing sarcoma cell lines, with increasing concentrations of DFMO, we see decreasing rates of proliferation of the Ewing sarcoma cells. And this is a reversible process, because if we start treating the cells with DFMO and then wash away the DFMO, we can see that in the beginning, when DFMO is present, the cells don't proliferate. And when we wash the DFMO away, they resume their ability to proliferate again. So DFMO stops the cells from dividing, but it doesn't kill them in tissue culture, because we can wash the DFMO away and they'll start to divide again. This is a direct result of depleting polyamines from the cells, because if we do exactly the same experiment that I just described, but we include spermidine in the culture, the spermidine can diffuse into the cancer cells, and in the presence of spermidine, the cells will resume proliferating, even though DFMO stopped them from doing so. And DFMO continues to be present throughout this entire experiment. So for the first 4 days in this experiment, cells are grown with DFMO and they don't proliferate. And then we add spermidine, but keep DFMO around, and the cells start to proliferate again. And that's the proof that it's the depletion of the polyamines that makes the cells stop dividing, because I give the polyamines back and the cells start dividing again. And then we can show that it's at a particular phase in the cell cycle, which is kind of interesting, but probably not directly relevant to what we're looking at long term. I've not really just described to you a drug that sounds like it would be useful therapy, because it doesn't kill the cells, it only makes them stop dividing, and there are ways to work around that. But that's an artifact of studying Ewing sarcoma growing as individual cells on a flat piece of plastic in the rigidly defined conditions in our lab. So the next step we took was to look to see what happens if, instead of looking in 2-dimensional culture, we look in 3-dimensional culture at cells growing either as spheres because they can't attach to the plastic, or as colonies in soft agar, which is kind of like jello, that prevents them from attaching to anything but each other. And under those conditions, the control cells grow really nicely. So these are spheres, and these are colonies over here. But with relatively low concentrations of DFMO, they're completely abolished. No spheres, no colonies. So that points to the critical difference between studying cells growing in a three-dimensional culture model like this and a two-dimensional culture model like I showed you before. And it follows that if 3D culture is different than 2D culture, then looking at the impact in animals might be different than looking at the impact in cultured cells. And so the next thing we did, of course, was look in vivo in our mouse model. So using the mouse model that I described to you before, where we implant the tumor into the leg, let the tumor grow, amputate the leg, and see what happens afterwards. So the first thing we did was try to confirm that ODC activity differs between tumors that were put in the subcutaneous space and tumors that were put into the tibia, which follows from the work that we showed before. And this was a collaboration with this gentleman here, Bob Casero, who is a professor at Johns Hopkins and has been our generous supplier of DFMO for several years now, which is great because the market value, the retail value of the DFMO that he has given to us over the last 5 years, calculated once, is somewhere close to $1 million, and we haven't had to pay for it, which is kind of. So we were able to show in the tumors that were implanted in the leg that their ODC level was actually less than the subcutaneous tumors, which matches with the difference in arginase level that I showed you before. And coincident with the lower level of ODC activity is a lower level of the polyamines themselves, putrescine lower in the orthotopic tumors than in the subcutaneous tumors, and spermidine lower in the orthotopic tumors than the subcutaneous tumors. So that's just in tumors growing in mice. We haven't introduced DFMO yet. We wanted to make sure that in line with what our initial observations, that ODC activity and polyamine levels are different in the subcutaneous tumors compared to the orthotopic tumors. So then we look to see what happens if we give the mice DFMO. So DFMO is a great drug to give to mice because unlike drugs that have to be given by injection that are labor intensive, or drugs that have to be given by putting a feeding tube into the esophagus of the mouth, which is also super labor intensive, DFMO is dissolvable in water, and so we can just dissolve it in their drinking water and let them drink as much DFMO-laced water as they want, and that makes it easier to do these experiments, which makes the people who actually do the work really happy. And then what makes me happy is that the results matched what we had expected. So we were able to show that the tumors isolated from mice that were given DFMO in their drinking water have less ODC activity than control mice that don't have DFMO in their drinking water. So we were able to show that adding DFMO to the drinking water of the mice inhibits the target enzyme even in the tumors growing in the mice, which is important if this is going to be a drug one day. And we can see an impact of that by looking at polyamines that are secreted in the urine of the mice, where there's a decrease in both putrescine and spermidine in the urine from the mice. And that's important in thinking about translating this into a trial for humans, because it's easy to monitor whether the drug is doing what it's supposed to be doing if we have a urine test, which is even easier than a blood test, because kids sometimes protest blood tests, but I've never had a kid refuse to pee. We can certainly outlast the stubborn ones who try. We also looked at polyamine levels in the tumors themselves and showed that in the mice that were given DFMO in their drinking water, there was much less putrescine in the tumors than in the control mice, and same with spermidine, and a little bit of a difference in spermine. So DFMO given in the drinking water penetrates all the way into the tumors where it inhibits ornithine decarboxylase, thereby decreasing the level of polyamines both in the tumors and then to be secreted into the urine of the mice, all of which allowed us to then ask the question of whether this impacts the way the Ewing sarcoma model behaves in the mice. So going back to how the experiment is set up, tumor is implanted into the tibia of the mouse, we let the tumor grow, then we do surgery to amputate the limb, and the data that I'm going to show you involve giving DFMO to the mice starting at that point. So the primary tumor is gone. but there are cells that have disseminated through the mouse's body, and we can prove that in some of our experiments by isolating lungs from the mice, and from the mouse lungs, we're able to isolate human Ewing sarcoma cells. So we know that at the time that we do the surgery, the tumor cells have already disseminated. And at this point, we change their drinking water to include DFMO, and we follow the mice to see what happens to them as they live or don't. And what we found was really, really cool, I think. So this is the survival curve for the mice, and the red line is the mice that were given control regular drinking water to drink, and you can see that around a month or so after amputation, they start to develop metastatic disease and die. And the mice who were given DFMO in their drinking water, none of them died of metastatic disease. So the cohort isn't huge, but the p-value is significant, and the observation that none of the mice developed metastatic disease is kind of cool, or at least metastatic disease enough to die from. When we did necropsies, so like an autopsy but for mice, we see a big difference in what the metastases look like. So panel B is a metastasis that spread to the kidney of one of the control mice, and you can see that there's a smidge of kidney tissue hiding back here, and a little bit here, and a little bit here, but all the rest of this is metastatic cancer. These are the two of the only metastases that we saw in all of the DFMO treated mice. And you can see this is also in the kidneys, but you can see that they're these little tiny dots, not these big tumors that are destroying the kidneys, which is why this mouse didn't die from metastatic disease. When we look under the microscope in the control mice, we see very healthy looking tumors. Here's an example of one. And to prove that this is a human tumor and not a spontaneous tumor that happened to arise in a mouse that was involved in our experiment, we stained the tumor for NKX 2.2, which is a human-specific marker of Ewing sarcoma, and you can see that it stains the tumor nicely here. So this is a nice, healthy Ewing sarcoma metastasis in a control mouse. This is a slice through this tumor right here, and you can see two things. So the first is this really looks like dead goop, not a healthy tumor. Compare this picture to that picture. And when we stain with NKX 2.2, we do see a little bit of staining to prove that the cells that are there and haven't died yet are human Ewing sarcoma cells, but compare this with this and compare this with this, and you can see that there's a profound impact of DFMO on even what happens to the metastases that are able to develop. And in order to quantify this kind of difference, we developed a measure that we call a metastatic index. So what a metastatic index is for each mouse, we count metastases, and you get 1 point per metastatic lesion. So if you have 2 lesions like this mouse, because these 2 kidneys were from the same mouse, that's 2 points. Each metastasis is also graded based on its size. A small metastasis gets 1 point, a medium-sized one gets 2, and a large one gets 3. And so so a large metastasis is 3 points, a small metastasis is 1. You add those up per mouse, and you get a metastatic index, and the control mice are here, and the DFMO treated mice are here. And you can see that there's a very big difference in the extent of metastatic disease between the control mice and the DFMO treated mice. And these two that aren't 0 are these which are really dead. So if I really wanted to skew the data by only applying metastatic index to viable healthy tumors, these 2 dots would be down here, and the difference would be even more profound than it already is. So we wanted to put all of this together and understand, oh wait, I forgot to say that this animal experiment was done by this gentleman. This is Paul Sierro, who was my lab manager for 5 years and a huge San Antonio Spurs fan, even though he grew up in Rochester, so I don't quite understand that. But, we wanted to understand not just that DFMO has this effect, but how DFMO has this effect. And that was particularly important because all of the in vitro data that I showed you with the cells growing in tissue culture plastic showed that DFMO didn't actually make the cells die. And here I'm showing you that in the mice, it does make the cells die. So how does that work? Well, the first thing we did was look to see whether DFMO can cause apoptosis, which is the most intensely studied programmed cell death in the cancer field. And this is etoposide, which induces apoptosis strongly. And this is multiple different concentrations of DFMO, none of which induce any apoptosis at all. So the cells are dying, but it's not by apoptosis. How can that be? Well, that's because apoptosis is only one form of programmed cell death, and there are lots of other forms of programmed cell death. So we decided to try to figure out if DFMO is somehow causing a different kind of programmed cell death in order to have the effect that I just showed you. And we came across this paper that did show a link between polyamines and a type of programmed cell death called ferroptosis. So this is a figure from their paper that shows that P53, which is a really important cancer gene, can control the expression of an enzyme that they called SAT1. And then through some black box mechanism that they didn't investigate, they showed that SAT1 can regulate the expression of this enzyme called ALOX15. And ALOX15 is one of the key mediators of cell death caused by ferroptosis. And SAT1 is the same thing as this enzyme here, SSAT, which stands for spermine and spermidine acetyltransferase. And what this enzyme does is it modifies spermine and spermidine by adding an acetyl group to the end. And this form and this form are exported from cells and don't accumulate in cells. So SAT1's job is to kick polyamines out of cells. And so this model would suggest that kicking polyamines out of cells will activate ALOX15, which will activate ferroptosis. So we looked to see whether DFMO can affect ALOX15 expression in Ewing sarcoma cells. And obviously, I'm telling you this story so you know that the answer is yes. We're able to show in 2 out of the 3 Ewing sarcoma cells that we looked, cell lines that we looked at in vitro, that ALOX15, that DFMO increases the expression of ALOX15 either 2-fold or 7-fold in this case. And this is a direct consequence of the depletion of polyamines from the cells. Because if we do exactly the same experiment, but we include spermidine, so again, the spermidine diffuses into the cells and accumulates there and therefore bypasses the impact of DFMO on polyamine accumulation. When we do that, DFMO no longer has an effect on ALOX15 expression. So DFMO treatment of Ewing sarcoma cells increases ALOX15 expression directly as a consequence of its impact on polyamine accumulation. So this made us really excited to think about ferroptosis. And so we need a brief lesson on how ferroptosis works. So ferroptosis involves polyunsaturated fatty acids, which are a component of the plasma membrane of all cells, including cancer cells. And when polyunsaturated fatty acids are made into phospholipids, they accumulate in the cell membrane, and they can be peroxidated by an enzyme called ALOX15 that I was just telling you about, and another enzyme called PTGS2. And through metabolic pathways that include glutathione and amino acids like cysteine, and also iron metabolism, that's where the name ferroptosis comes from. Iron is involved, and so is cholesterol synthesis, that's over here. Through an interplay of all of these pathways, peroxidated phospholipids that contain polyunsaturated fatty acids accumulate in cells. They cause the cell membrane to rupture, and that is how ferroptosis works. So we tried to look a little bit more carefully into ferroptosis in Ewing sarcoma cells to see if that's what's actually happening in tumors that are treated with DFMO. So the first thing we did was an RNA sequencing analysis comparing primary tumors in mice with disseminated tumor cells to see whether the process of spreading away from the primary tumor changes anything about the cells that would be relevant to this pathway. What we found was that In the disseminated tumor cells compared to the primary tumor, there's downregulation of glutathione metabolism, and glutathione is a key inhibitor of ferroptosis. So disseminated tumor cells have less glutathione metabolism, specifically the enzymes in the GST pathway that are critical for using glutathione to oppose oxidative things that happen in cells. So that's less in the disseminated cells than in the primary tumor. We also saw downregulation of an enzyme called ACSL3, and ACSL3 mediates the degradation of polyunsaturated fatty acids. So if there's less of this enzyme, you would predict that that would mean there would be an accumulation of polyunsaturated fatty acids, which would increase a cell's vulnerability to ferroptosis. We also saw an increase in the expression of this enzyme, which can directly mediate the peroxidation of polyunsaturated fatty acids, which is the last step before the cells die. And so putting these things together, our conclusion was that the cells that have disseminated away from the primary tumor, in other words, the ones that are going to become metastases, display an increased vulnerability to ferroptosis compared with the primary tumor. So then we looked at the metabolism of the primary tumors and the disseminated tumor cells. And what we found was increases in cysteine and cysteine glutathione disulfide and decreases in glutathione itself that would also be predicted to increase the vulnerability of these cells to ferroptosis, because glutathione is one of the major things that counteracts the peroxidation of the polyunsaturated fatty acids, which again is what drives the cells to die. Finally, none of the stuff that I just showed you has anything to do with DFMO. Finally, we asked the question, what does DFMO do? Unfortunately, since when we treat mice with DFMO, we don't collect metastases that we can evaluate because the DFMO is doing its job in preventing the metastases from happening. We had to analyze the primary tumor either from control mice or mice that were treated with DFMO. But when we did that, we found a decrease in cell cycle genes and a decrease in apoptosis genes, which fits with what we found in vitro, that DFMO makes the cells stop dividing, but doesn't induce apoptosis. We also found a decrease in this gene, HSPB1, which is a strong negative regulator of ferroptosis. So decreasing a negative regulator is something that would activate that process. So this is something that would activate ferroptosis. We see decrease in HMG-CR, which is the rate-limiting step in cholesterol biosynthesis. This is also a negative regulator of ferroptosis. It's downregulated by DFMO, so that would have the net effect of activating ferroptosis. We see an increase in the enzyme LPCAT3, which is a driver of the conversion of polyunsaturated fatty acids into phospholipid polyunsaturated fatty acids, which is a necessary component of ferroptosis, and that's also increased by DFMO. Then looking back at our metabolic data, we were able to show that DFMO increases the accumulation of polyunsaturated fatty acids in Ewing sarcoma cells compared to monounsaturated fatty acids, showing that what we're seeing is not just an increase in the presence of enzymes, but also their byproducts, the polyunsaturated fatty acids that get peroxidated in order for the cells to die. So putting all of that data together, we see in cells that have disseminated away from the primary tumor, metabolic and gene expression changes that are predicted to increase vulnerability to ferroptosis. And then in cells that have been exposed to DFMO, a series of gene expression changes that would be expected to trigger ferroptosis. And the end result is ferroptosis of the disseminated tumor cells specifically, thus a lack of the development of metastasis, thus survival of the mice. So the conclusion of all of this preclinical work is that when Ewing sarcoma cells disseminate, they become sensitized to undergo ferroptosis due to changes in gene expression associated with lipid metabolism and glutathione metabolism. And then DFMO, by depleting cells of polyamines, upregulates genes that drive the production of polyunsaturated fatty acids and genes that are executioners of ferroptosis, like ALOX15. And so we believe that induction of ferroptosis in the disseminated tumor cells specifically is the mechanism by which DFMO prevents metastasis to happen in mice that have already had tumor cells disseminated away from their primary tumor. So how is this going to work when we move on to humans? Because we, of course, do all of this work to try to develop better ways to treat kids with Ewing sarcoma. So that leads to our current thoughts about a clinical trial of DFMO in kids with Ewing sarcoma. And the key point in thinking about designing a clinical trial of this compound for these patients is that DFMO works by preventing the disseminated tumor cells from becoming metastases. In data that I didn't show you because I already talked too long, the DFMO treatment actually doesn't really significantly impact the growth of the primary tumor in our mouse models. And so it might not affect the growth of metastases that you can see on a chest X-ray or on a CAT scan, because they may be more like the primary tumors. And really, we're trying to target disseminated tumor cells that haven't become metastases yet, so that they won't become metastases and our children will live longer. The other thing to think about is that there are already effective chemotherapy regimens for relapsed Ewing sarcoma. So if we were developing a trial for kids with relapsed Ewing sarcoma and proposed treating just with DFMO, it might be hard to convince parents to enroll their children in such a trial if that meant bypassing chemotherapy treatments that are better established that we know actually help some patients already. So our target population is patients with relapsed Ewing sarcoma, and the design is to offer those patients a standard chemotherapy regimen that's anyway offered to kids with relapsed Ewing sarcoma, cyclophosphamide and topotecan, but to have a randomization on top of that to either get DFMO or not get DFMO, to see if the addition of DFMO will improve the outcomes compared to cyclophosphamide and topotecan alone. And luckily for us, this regimen, cyclophosphamide, topotecan, and DFMO, has actually already been tried in kids with neuroblastoma. So we know it's safe. We know we can give these drugs in this combination to children with cancer and it won't hurt them. And so that allows us to sort of bypass the first stages and just go straight to the money, which is the trial that we want to do. Our primary outcome is going to be event-free survival, specifically focusing on the prevention of new metastases rather than necessarily the change in the metastases that you can already see. And the idea would be that the cyclophosphamide and topotecan, we think, does a reasonable job of targeting the metastases that we can see, but not so much the metastases we can't see. That's why cyclophosphamide and topotecan isn't curative for most kids. But maybe if cyclophosphamide and topotecan can target what we can see and DFMO can target what we can't see, that combination will be much better than either one alone. And then part of the reason for going through all of the biomarker stuff that I did before is because we can use those as markers to make sure that the DFMO that we're giving to the kids is actually hitting its target by checking for polyamines in their urine. And then we also thought about ways that we can look to measure peroxidation of lipids in circulating tumor cells, since if our hypothesis is right, that DFMO prevents metastasis by triggering ferroptosis, we ought to be able to see evidence of that in cells that have disseminated away from the primary tumor. So this trial model was literally developed yesterday. And so that's one of the reasons that I'm interested in feedback from this group, because I want to think about proposing a trial that is not just interesting and exciting to the clinicians and scientists, but also attractive to parents. And so with that, I will stop and show my acknowledgments, including the folks who did this work while I was still at Hopkins, our collaborators at Hopkins, the people who have helped us with our RNA sequencing and metabolomics analyses, all of our current lab members at Einstein who are pictured here at our recent lab picnic, the people who have funded our work, and I am happy to transition to the Q&A section.
Great.
Well,
Thank you so much. That was fascinating, to say the least, and really well presented in a way I think many of us could understand. This neuroblastoma space in the DFMO, we, as Little Warrior Foundation, have become very close with the organization that is funding a lot of that research. We'll be talking to Dr. Scholler in a couple hours here. And I encourage anyone who hasn't heard the story of how DFMO kind of came to be and how it's impacted the world of neuroblastoma to look into it. It's like a made-for-TV movie coming soon too. So with that in mind, people are asking in the chat bot how DFMO is administered, in the neuroblastoma space, or how you envision it for the trial, and what are the side effects for DFMO and its impact on healthy cells?
Great question. So DFMO is an oral drug, and so it actually comes in both a capsule formulation and as a liquid. And so we hope it will be relatively easy for Ewing sarcoma patients. Since most Ewing sarcoma patients are the appropriate age to be able to take pills, the liquid is less important. That's kind of important for the infants and toddlers who get neuroblastoma. But it's an oral drug, which means that you don't have to come to clinic to get treated. You can just take your pills at home, which is kind of nice. Side effects, just like with everything else, vary with dose. With the dose that the FDA seems to be poised to approve for neuroblastoma, there are actually very few side effects, and it's really well tolerated, which is why it's easy to envision a trial combining DFMO with chemotherapy, because chemotherapy is bad, but if you add something that's relatively non-toxic on top of the chemotherapy, then it at least won't make it that much worse. And if this is a drug that might ultimately need to be given for a prolonged period of time, it's nice to be able to give something that's really well tolerated. There's a little bit of nausea associated with it, but I'm told not overly horrible. When it's given at higher doses, and some of the neuroblastoma trials have involved significantly higher doses than what was proposed to the FDA, there is hearing loss associated with it, and that's kind of a big deal because, especially in the neuroblastoma world where patients are also treated with platinum agents that cause hearing loss, the combination of chemo that causes hearing loss plus a maintenance drug that causes hearing loss isn't quite as attractive as something that doesn't have sort of that overlapping toxicity. We are hopeful that by monitoring biomarkers like urinary polyamines and peroxidation of lipids in circulating tumor cells, we'll be able to show that we're hitting the target, if you will, with the doses that are closer to the ones that are about to be approved by the FDA that don't have hearing loss as an associated side effect. If not, and we have to go up to higher doses, that's something that we'll have to think about how to manage, and just at least be thankful that the chemotherapy that kids with Ewing sarcoma get doesn't include cisplatin or anything else that that causes hearing loss, and so maybe this won't cause. It would be terrible to put parents in the position of having to choose between hearing loss and relapsed cancer. My guess is most would favor hearing loss that can be addressed with a hearing aid over relapsed cancer that is much harder to address. But if we can stay away from those doses and stay at the doses that don't do that, that would obviously be better.
Great.
Yeah, I would say, go ahead, Amy.
I would say, following on with that, do you envision DFMO as a possible maintenance therapy for someone who has achieved no evidence of disease?
So that was actually our original plan. So prior to yesterday, I would have shown you a clinical trial design where we took kids who were diagnosed with metastatic Ewing sarcoma, completed their standard upfront therapy, and ended up with no evidence of disease at the end of treatment, and gave DFMO to those patients. And in my mind, that's the cleanest way to answer the question of whether DFMO does what does in humans, what we've shown it does in mice. And the reason that we started thinking a little bit differently is that we think that within the time frame that this trial would be running, COG is going to be opening another study for newly diagnosed patients with metastatic Ewing sarcoma. And if they do, that those patients would not be eligible for our study. Nobody would forgo the new COG study because maybe they would achieve remission and at the end be able to do maintenance therapy. Or maybe I'm overstating it and not nobody, but it would impact on our ability to enroll patients and thereby slow up the process of demonstrating in humans that DFMO helps. I think that it would be different if we had human data on DFMO in Ewing sarcoma, because then we could propose something different already. This would be the first clinical trial of DFMO in kids with Ewing sarcoma. And so the first step is to come up with a trial design where we can see an impact of DFMO in a patient population that doesn't have a whole lot of other decent options.
I guess I will push back. Sorry.
Well,
Dr.
please
Loeb.
do.
I'm going to push back, and I'm going to make sure that the patient population answers your question, like who wants to see this type of clinical trial. So please throw your comments in so Dr. Loeb can hear that loud and clear. But I guess I would push back and say, let's not let this be a rate limiting step, right? Like regardless of what COG is going to do, COG is going to do it. But if you clearly have something that's novel and can prevent relapse, I think the sooner we get there, the better for everybody. So I'm gonna let the patient's perspective speak for that. And that's just my take right now. The one question I do have for you is there any evidence that says that DFMO can cross the blood-brain barrier? Or do we not know that?
Wrong pronoun. I don't know that. 15 minutes with the internet and I can get
Yes.
you an answer to that
Yeah,
question.
that's all right. That's all right.
I honestly don't know, mostly because I haven't, our mice don't develop brain metastases. So in our lab, we just haven't had occasion to answer that question. In the adult world, the focus is also on tumor types that don't necessarily metastasize to brain. And so I don't know the answer to your question.
Okay, fair enough.
But that doesn't mean somebody doesn't know it.
Right. Do you want to take the next question? We got a couple minutes here.
Yes. So there are a lot of great questions. So would this treatment would begin after frontline has been completed and then we've experienced a relapse, correct?
Well, it depends on what the end trial design ends up being. It's definitely in the beginning, the idea would be to use this to prevent relapse. And the operative question, I guess, is prevent any relapse or prevent another relapse. So if in the first trial design, not the one in the slides, but the one that was the earlier idea, that the idea would be to start the clinical testing of DFMO in a population of patients who don't have radiographically evident disease, but are at very high risk of developing relapse. So that's the rationale for choosing patients who were initially diagnosed with metastatic disease, because we can get those patients into remission, but we know that if the cure rate is only 20% or so, that means 80% of those patients are going to have a relapse. And so that's a patient population at very high risk. And so that's a place where it would be easy to show, relatively easy to show, that DFMO can prevent relapse. We're still going back and forth about what the trial design would look like. The one that I described to you would more be designed to prevent another relapse, because it would be taking patients who have relapsed Ewing sarcoma, giving them standard therapy for relapsed Ewing sarcoma, and adding to that DFMO. And I would envision, if the DFMO part is well tolerated, extending that beyond the time that the patient is getting chemotherapy, with the goal being the DFMO component of that treatment is designed to prevent the next relapse. Because we know that patients who have one relapse, even if we can get them back into remission, are very high risk of developing a second relapse and then a third relapse and so forth. And so the goal would be, if that ends up being the trial design, using DFMO to prevent any further relapses. And I think I've heard good arguments. I hear a good argument right now for my original idea, heard a reasonable argument yesterday for the idea that I presented here, open to hearing other arguments in favor of either one of those things. But really what we want to do, the sort of expedient goal is to come up with a design of a trial that we can open as soon as humanly possible, that can accrue patients as quickly as possible, so that we can see in as short a time as possible if DFMO helps or doesn't help. If it does help, then it becomes much easier to incorporate that into the kind of trials that we would really like to do. Like, I could envision a trial for patients newly diagnosed with metastatic Ewing sarcoma, where the intent of the trial from day one is standard chemotherapy with surgery and radiation and whatever, and immediately upon completion of all of that, transition to DFMO and just take DFMO for a couple of years and see if that prevents those patients from ever having a relapse. It's hard to justify to the people that sort of do the regulatory and accounting pieces in our world a trial like that with zero evidence that DFMO helps treat Ewing sarcoma in a human, just based on our mouse data. So that's why that's not the first step. But once we can show that DFMO can prevent relapse, not just in mice, but also in humans, then it's much easier to convince people to do the trial that I imagine the parents really want, which is the one that I just described, where the DFMO starts as soon as therapy ends in patients who are at high risk and prevents them from ever having to have a relapse in the first place.
Yeah, I think there's a lot of comments here saying do it all and whatever it takes to get to the clinic as fast as possible. People are appreciative of it. To me, it seems like we have low-hanging fruit. I love that your focus is on relapse and metastatic disease, but we know that 25% of those with localized disease will, for some reason, relapse. So, I mean, I think there's an arm for that as well. But I hear you loud and clear. One last question is, why cyclotopo and not IT? Was that purely because of the work done in neural and human? Is that why you chose that regimen?
Yeah, essentially. If we end up having to do something where there's a period of time on the study where patients are given both a chemo regimen and DFMO, then we're going to have to, at some point, prove that it's safe to do that. And we can bypass the it's safe to do that part if we have data from another disease type, like neuroblastoma, where we already know that it's safe to do that. So if I propose a totally novel combination, irinotecan temozolomide plus DFMO, to the local IRB, or it wouldn't be one local IRB, lots of IRBs, the first thing the reviewers are going to say is, how do you know that that's safe? And I won't be able to give them an answer, so they're going to reject it unless we come up with some kind of lead-in study that requires probably dose escalation of DFMO from a low dose that's less likely to help before we get to a higher dose that's likely to help. And so what that means is that the kids that enroll in that study first are going to get a lower dose of DFMO, and maybe that dose of DFMO is less likely to help them. So those kids are not getting the same opportunity of cure as the kids who join later when we've been able to escalate the dose. So if we have a regimen where we already know that in neuroblastoma therapeutic dose of DFMO is safe to combine with the chemotherapy, we bypass those earlier steps and go straight to what we really want to do, which is give as much DFMO as is safe to do in a context where we're not going to hurt anybody.
Okay. I hear you loud and clear. I think there's a lot of appetite from the patient community, and even at Little Warrior here too. If we can help expedite things, help with patient accrual, regardless of what COG does, we are always looking for fast moving ships that have sound preclinical evidence or in-human evidence. I think there's plenty of in-human evidence to move forward with a trial like this. So we're going to wrap it up because we are at the hour mark. I know we could probably talk forever and ever, but I want to thank everybody for being here. Dr. Loeb, thank you for being here, and thank you for that master class in the biochemistry of tumor biology. Like, it's obvious to me that you're a brilliant scientist as well as a great teacher, like that we learned a lot in a short amount of time. And lastly, from the greater Ewing sarcoma community, thank you for making our kids' lives a priority in your work. Thank you to my fabulous co-host and co-founder, Emily, for helping me moderate the conversation. And lastly, thank you to our viewers for joining us today here as well. For more Ewing's U, we'll see you back here again in 2 short weeks on November 30th at 2 p.m. Eastern. We're going to go to St. Jude's and with Dr. Sarah Federico to talk about 2 unique and novel clinical trials they have there. So please join us for that one. And as always, feel free to find us on the more information about our webinar series at Ewing's U. You can do that by going to littlewarrior.org, navigating to the Ewing's U tab on the website. Also follow us on Instagram and Facebook, as we always post about our next speaker that way as well. Lastly, and in closing, we have a sign off here at Little Warrior that embodies our fights in honor and in memory of so many little and big warriors who have faced this cancer. It's how we sign off on all our communications, both internally and externally. Dr. Loeb, on a count of 3, it's just simply a swords up. That's how we sign off. If you could join us, greatly appreciate it. Thanks everybody for being here. 1, 2, 3.
Swords up.
Swords up, everybody. Thank you. We'll see you soon.
Thanks. Bye, everybody.